Hi, I'm Brian, and this is cellular respiration and photosynthesis. Let's start with cellular respiration. Now, you might be wondering, what is cellular respiration? Well, cellular respiration is a series of chemical reactions converting stored energy into nutrients so the cells are ready to use. In other words, it's just stored energy converting nutrients for the cell to be able to do work. In this, there are 5 stages. Stage 1, glycolysis. Stage 2, acetyl-CoA synthesis. Stage 3, the citric acid cycle, and Stage 4, oxidative phosphorylation. In glycolysis, it is basically just the breakdown of glucose to a pyruvate. It means splitting sugar because since glucose is a 6-carbon sugar, it splits into 2, having 2 3-carbon molecules. This has 10 chemical reactions but can be divided into 3 phases. In the first phase, reactions 1 to 3, it prepares glucose for next stages by adding 2 phosphate groups to produce fructose 1,6-biphosphate. To even do this step, 2 molecules of ATP are hydrolyzed per molecule of glucose. When phosphorylated glucose is trapped inside the cell, 2 negatively charged phosphate groups destabilize the molecule, so it can be broken down in the next phase. In the second phase, reactions 4 to 5, in reaction 4, the fructose molecule splits into 2 3-carbon molecules, glycerol 3-phosphate and its isomer dihydroxytoluene phosphate. In reaction 5, dihydroxytoluene phosphate is converted into glycerol 3-phosphate. For each glucose molecule entering glycolysis, 2 3-carbon glycerol 3-phosphate molecules enter phase 3. In the third phase, reaction 6 through 10, it ends with the production of 2 molecules of a pyruvate.
It starts with 6-carbon glucose, and the body injects energy in the form of phosphate into this molecule so that it can divide it into 2 3-carbon sugars. It's very similar to using an axe to split a log to make firewood. So the result are these 2 3-carbon sugars. They're a bit different. On the right is dihydroxyacetone phosphate, which can go through glycolysis or onto lipid metabolism. The guy on the left can go directly into glycolysis. Grab the poster if you need to memorize this. If you're just interested in metabolism, this is a fundamental metabolic pathway. The end result of it is a molecule called pyruvate.
In the second stage of cellular respiration, which is called acetyl-CoA synthesis, this takes place in the mitochondria matrix. The pyruvate is transported into the mitochondria matrix, which is converted to acetyl-CoA. The pyruvate is oxidized and splits to form carbon dioxide. Electrons are donated to NAD+, which then turns into NADH. The remaining pyruvate molecule is transferred to the coenzyme A that carries the acetyl group to the next set of reactions. Overall, it is just the synthesis of 1 molecule of acetyl-CoA from pyruvate to form 1 molecule of carbon dioxide and 1 molecule of NAD.
First, one of the pyruvates is oxidized, which basically means that it's combined with oxygen. One of the carbons off the 3-carbon chain bonds with an oxygen molecule and leaves the cell as CO2. What's left is a 2-carbon compound called acetyl coenzyme A, or acetyl-CoA. Then another NAD+ comes along, picks up a hydrogen, and becomes NADH. So our 2 pyruvates create another 2 molecules of NADH to be used later.
Of cellular respiration, which is called the citric acid cycle, but can also be called the Krebs cycle and the tricarboxyl cycle. This takes place in the mitochondrial matrix as well as the synthesis of acetyl-CoA. Composed of 8 reactions like a cycle because oxalose acetate starts out first and is regenerated at the end of the cycle. First, a 4-carbon molecule of oxaloacetate reacts with the 2-carbon acetyl group of acetyl-CoA to form a 6-carbon molecule of citrate. Then 2 carbons are eliminated because there are 6 carbon molecules and are trying to get 4. Later, the reactions produce electron carriers. 3 molecules of NADH and 1 molecule of FADH are produced per turn of the cycle. They carry high-energy electrons to power the synthesis of ATP.
Citric acid is then oxidized over a bunch of intricate steps, cutting carbons off left and right to eventually get back to oxaloacetic acid, which is what makes the Krebs cycle a cycle. And as the carbons get cleaved off the citric acid, there are leftovers in the form of CO2 or carbon dioxide, which are exhaled by the cell and eventually
in the
by
fourth
you.
stage of cellular respiration, which is called oxidative phosphorylation. In this stage, electron carriers generated in steps 1 to 3 donate high-energy electrons to an electron transport chain. An electron transport chain is a system that transfers electrons along the membrane filled with proteins to a final electron acceptor, releasing the energy to form ATP. The electrons donated by NADH entered through complex 1 and electrons donated by FADH entered through complex 2. They entered through complex 1 or 2 to complex 3 and then move 4. In this stage, ATP synthase is formed. ATP synthase is an enzyme that moves protons to the enzyme of the synthesis of ATP.
These proteins will swap these electrons to send hydrogen protons from inside the very center of the mitochondria across its inner membrane to the outer compartment of the mitochondria. But once they're out, the protons want to get back to the other side of the inner membrane because there's a lot of other protons out there, and as we've learned, nature always tends to seek a nice peaceful balance on either side of a membrane. So all of these anxious protons are allowed back in through a special protein called ATP synthase. And the energy of this proton flow drives this crazy spinning mechanism that squeezes some ADP and some phosphates together to form
Overall,
ATP.
each of these steps are accompanied with the enzyme phosphofructokinase 1. It is an enzyme that regulates the rate of glycolysis. It has many activators and inhibitors which allows it to control the movement of glycolysis. Its ATP levels are low, then it activates allowing glycolysis to continue. But if ATP levels are high, it inhibits and slows down glycolysis. This is the formula for cellular respiration. It says that C6H12O6 plus 6O2 yields 6CO2 plus 6H2O plus energy. In other terms, it's saying that glucose plus 6 molecules of oxygen make 6 molecules of carbon dioxide with 6 molecules of water with energy, or in other words, ATP. In this video, they show us the chemical formula of cellular respiration. We already know what the reactants and the products are, but if you switch the chemical formula around and replace energy with sunlight, you will get sunlight plus 6 molecules of water plus 6 molecules of carbon dioxide makes 6 molecules of oxygen and glucose, which is the chemical formula for photosynthesis. As you can see, both cellular respiration and photosynthesis share the same products and reactants, just opposite of each other. Now we are on photosynthesis, and you might ask again, what is it? Well, it's a biochemical process where carbohydrates are built from carbon dioxide and energy of sunlight to turn into glucose and oxygen. It's a redox reaction, which means that the oxidation number of a molecule changes by gaining or losing an electron. In photosynthesis, water is being oxidized, which means that it loses electrons, which is called oxidation, and carbon dioxide is being reduced, which means it gains electrons, which is called reduction. It all begins with photosystems and the photosynthetic electron transport chain, and ends with the Calvin cycle. In photosynthesis, there's something called the photosynthetic electron transport. It connects to photosystems, which are photosystems 2 and photosystems 1. Photosystems 2 supply electrons to the beginning of the electron transport chain. It pulls electrons from water. Cytochrome B6F complex carries electrons between photosystems 2 and photosystems 1. Plastiquinone carries electrons from photosystem 2 to the cytochrome. Plastocyanine carries electrons from cytochrome to photosystem 1 by diffusing through the lumen.
PS2's chlorophyll now has this electron that is so excited that when a special protein designed specifically for stealing electrons shows up, the electron actually leaps off of the chlorophyll molecule onto the protein, which we call a mobile electron carrier because it's a mobile electron carrier. Splits that ultra-stable molecule, H2O, stealing one of its electrons to replenish the one it lost. The byproducts of this water splitting, hydrogen ions, which are just single protons, and oxygen. Sweet, sweet oxygen. This reaction, my friends, is the reason that we can breathe. Pumping the thylakoid full of protons, we're creating a concentration gradient. The protons then naturally want to get the heck away from each other, and so they push their way through an enzyme straddling the thylakoid membrane called ATP synthase. And that enzyme uses that energy to pack an inorganic phosphate onto ADP, making ATP the big daddy of cellular energy.
Next, we have the cyclic electron transport. This video will show you how the process of it works.
In cyclic electron flow, we're not going to use photosystem 2 at all. We're only going to use photosystem 1. So photosystem 1 will absorb light and use that energy, capture it as high energy electrons, which pass it all the way to the P700. And now we have a high energy electron here. Okay? Goes to the primary electron acceptor to ferredoxin as before. But unlike before, ferredoxin is not going to give that electron away for NADPH. It's going to actually go back and give it to plastoquinone instead, to the beginning of the electron transport chain. So
as you can see in the photo, it only wants to produce ATP and prevent reduction of NADP+.
So when the electron gets to plastoquinone, it's going to go down the electron transport chain as before, releasing its energy bit by bit as it actively transports hydrogen ions across the membrane. And it goes all the way back to photosystem 1 again. And from here we can do it all over again.
Occurs in photosystem 1, and ATP is the only thing that's produced out of it. It stores its released energy as a proton gradient. The energy provided to produce ATP comes from the redox reactions of the electron transport chain. Electrons from photosystems 1 are redirected from ferredoxin back into the electron transport chain. This prevents reduction of NADP+ and produces only ATP. Next we have is the Calvin cycle. It consists of 15 chemical reactions to synthase carbohydrates that can be grouped into 3 groups: carboxylation, reduction, and regeneration. Carboxylation, carbon dioxide added to a 5-carbon sugar called ribulose 1,5-bisphosphate, or RUBP, then catalyzed by ribusco, which is an enzyme. Then carbon dioxide and ribusco must diffuse into the active site. The product is a 6-carbon compound that breaks into 2 molecules of 3-phosphoglycerate, or 3-PGA.
This phosphate gets a CO2 slammed onto it, and then immediately the whole thing gets crazy unstable. The only way to regain stability is for this new 6-carbon chain to break apart, creating 2 molecules of 3-phosphoglycerate. And these are the first stable products of the Calvin cycle.
In reduction, for energy to increase, NADPH transfers energy and electrons to allow carbohydrates to be synthesized from carbon dioxide. Then ATP is used to phosphorylate 3-PGA, and NADPH transfers 2 high-energy electrons to the phosphorylated compounds. Since 3-PGA are formed each time ribusco catalyzes 1 molecule of carbon dioxide, 2 ATP and 2 NADPH are required for each molecule of carbon dioxide. The energy transfer steps result in the formation of glyceraldehyde 3-phosphate GAP and DHAP to form triose phosphate. And regeneration, it involves the regeneration of RUBP. It requires 2 molecules of NADPH and 3 molecules of ATP for each molecule of carbon dioxide. NADPH provides energy to the carbohydrate molecules, and ATP provides energy to regenerate RUBP. In photorespiration, it is almost the same thing as the Calvin cycle, but there's a difference. The Calvin cycle starts with carbon dioxide and ribulose, but the photorespiration uses oxygen and ribulose. And you can see as photorespiration, carbon dioxide is seen taken out of the cycle because the conversion of 2-phosphoglycerate into 3-PGA results in the net loss of reduced carbon.
Just notice that when oxygen is incorporated instead of carbon dioxide leaves the cycle, meaning we lose carbon from this cycle when we incorporate oxygen. So photorespiration results in the loss of carbon. This is a problem because the whole point of the Calvin cycle is to bring carbon in.
What he's saying is that this is basically the Calvin cycle except with the addition of oxygen.
Now, photorespiration is when ribusco takes oxygen instead of carbon dioxide and fixes that into the cycle. We call it photorespiration because it's part of the photosynthetic overall process, but it uses oxygen. So we think of that as respiration, right?